Hello everyone, I'm Claudia Colmo, a graduated master's student from Computation in Architecture at KDK. Today I'm presenting our paper, 3D printed biohybrid structures, a research that investigates the architectural potential of mycoremediation. Mycelium is the vegetative structure of the fungi and has the incredible ability to decompose toxic substances, filtering microbes, and therefore repairing the fragile territories and habitats. In the paper, we examine mycelium as a living remediator of contaminated sites. This land scenario posed significant challenges, being a source of natural habitat deprivation, as well as human occupancy impoverishment. We explored the concept of Mycorestoration into a speculative design proposition of a biohybrid system. The system uses soil as the raw material for construction and incorporates living mycelium using digital design and fabrication through additive manufacturing. We defined two investigations, one that is material with the 3D printing, one that is growth investigation, with the purpose to establish plausibility and test the feasibility of the concept, with particular focus of material composition, fabrication, and biocompatibility. The first investigation targets the composition of soil-based composites suitable for 3D printing, together with establishing process parameters to study compatibility, consistency, and repeatability. The 3D printing setup is composed by a UR5, 1 liter syringe capacity in which the material is inserted and pushed by air compressor, and a customized extruder with a nozzle of 7 millimeters. We developed successful soil composites that are biocompatible while extrudable. To improve the performance of the bulk material, we tested and incorporate various bioadditives. We produce two soil-based composites recipes, recipe A and recipe B, with embedded mycelium spawns. The primary parameters assessed for 3D printing are layer deposition height and linear deposition rate. And the combination of these two parameters set the quality of the material deposition. The second investigation seeks to set geometric and environmental parameters for promoting colonization of the mycelium and supporting its role as both structural binder and mycorestoration agent. This is explored through the synthesis of two printed prototypes, one skin section prototype and a double skin section prototype. The two prototypes are placed in a glass tank on top of a layer contaminated soil. And the two prototypes were grown in lab condition, and their development was observed over a time span. The internal volume of the section is filled with lignocellulosic substrate. The inoculation was made in non-sterile conditions with the Pleurotus austriatus spawns. For the one skin section prototype, the inoculation happens internally, and for this double skin prototype, the 3D printing is the method of inoculation, as the internal skin has embedded mycelium spawns. For the one skin prototype, the 3D printing is successful with compact walls. However, during the 30 days of incubation time, the mycelium does not produce a defined EFL network, and overall the growth does not result homogeneous. The 3D printing and growth investigation are both successful in the case of the one skin section prototype. In fact, over a period of time of about 35 days, the print was entirely colonized by the mycelium, and biocompatibility was established by the appearance of 3 generations of fruiting bodies. The mycelium created, in fact, a uniform network, and dense and abundant fruiting bodies are evident across many features of the print. Overall, the paper presents a speculative design concept of a 3D printed architecture comprising living fungal organisms to perform mycoremediation. The plausibility and feasibility of the two aspects of this concept, fabrication and growth, are investigated through preliminary experiments. However, questions regarding scalability of the system and practicality of achieving a continuous monolithic structure in open field condition remain open. Thank you very much.